Okay, it's time to get started. Thank you to those of you that have come back. I recognize some of you from the last session. So I am up again to speak to you. For those who may not have been in the previous session, I'm Laura Russell, and I'm the Training Officer for Community and Capacity at GBIF. Is that better? Okay. And my topic today is on the GBIF Capacity Development Framework. I will take you through the framework and provide some, but not all of the examples found in this document. At GBIF, we are in, I pressed the button. At GBIF, we are in the second year of our current 5-year strategic framework. This strategic framework aims to deliver enhanced value and coherence through continued innovation that further enables our community and helps our community meet its emerging needs. Priority 3 of this strategic framework aims to enable our network to meet future needs and challenges. This means we need to have the mechanisms in place to allow GBIF to help develop the capacity and expertise within our community. The Kunming-Montreal Biodiversity Framework, which is part of the theme of this session, in particular targets 20 and 21, which emphasize the importance of capacity building and the availability of biodiversity data, information, and knowledge, along with the UNESCO recommendation on open science, which emphasizes principles of inclusivity, transparency, and collaboration in scientific research, including the availability of open research data, provide an important global policy context for expanding capacity development actions through GBIF. As a result, during 2023, the GBIF Secretariat Community and Capacity Team redeveloped our capacity development framework to better define these mechanisms for our community development and to allow us to develop progress. The updated version was published earlier this year in March and contains 3 sections of objectives, implementation levels, monitoring progress, and then tools and approaches. So in the objectives section, we've simplified our capacity development work to 2 contributing main objectives. And these are, the first is to the capacity to develop and mobilize biodiversity data through GBIF, which includes supporting biodiversity data management and quality. The second is developing the capacity to use GBIF-mediated data. And this is to use the data both in scientific research and decision-making. In the second section, we target these objectives by working to develop capacity on nested levels from individual, institutional, national, and regional, and each with their own defined capacity goals. By working across these levels, we are embedding capacity in the layers of our network to strengthen the network as a whole, which ultimately supports the sustainability of the network. Additionally, within this second section, we have a series of metrics, some of which are shown here, on how each level contributes to our overarching objectives to mobilize and use data. In the third section of the framework, we focus on the tools and approaches in 4 specific areas. The first area is in supporting node development. Our nodes play an essential role in developing the capacity of institutions and individuals within their network and connecting to the national and regional levels, which means the nodes must also evolve their scope to keep pace with the changes within GBIF itself and in the broader biodiversity landscape. GBIF places strong emphasis on capacity development support for nodes through the exchange of experience and expertise. So since 2014, one of the key mechanisms for enabling collaboration and knowledge sharing between nodes is the Capacity Enhancement Support Program. GBIF, through core funds, administers the program in the form of co-funding for small projects between nodes targeting capacity exchange. The projects are based on a combination of mentoring, regional training workshops, and documentation. The program continues to receive a competitive number of project proposals each year, and the projects are essential in helping new nodes to get started and in strengthening partnerships between existing nodes. A second key mechanism in the support of nodes are the monthly technical support hours run by my colleagues in the data products team. So if you were in the last session before lunch, you will have seen Marie and Cecilia here. These are informal drop-in sessions, which start with short presentations exploring technical topics of relevance to the nodes, and then open for questions from the nodes. Topics have included things like the registry API, API SQL downloads, all very technical stuff. In the next area are our capacity development programs. GBIF has run various programs since its inception, with the scale and scope of these programs expanding over time, particularly in 2015 with the introduction of the Biodiversity Information for Development program. These programs are centered around open calls for proposals and combine other capacity development activities such as training workshops and webinars. These programs play an important role in developing capacities at the individual and institutional level, and help fund data mobilization as well as strengthen data management within institutions, data management capacity within institutions. During an independent evaluation of the BID program, the evaluation confirmed the increased availability of biodiversity data in the target regions and enhanced capacity to mobilize data. In addition, it found that BID contributed to the establishment of partnerships and that the projects achieved sustainability in a variety of ways, such as becoming a GBIF node, establishing new collaborations between participants in a variety of funding between BID projects and other funding initiatives, and the undertaking of dissemination activities to raise awareness on the benefits of open data access. The evaluation also provided practical recommendations that have informed our thinking on the next version of BID, and we know that the overall objectives will remain similar, but will have a specific emphasis on the need to implement targets 20 and 21. The third area in the tools and approaches is in support of the community of practice, which encompasses our GBIF volunteer community and our regional support teams. Volunteers play an essential role in the support of GBIF's programs and in strengthening skills across the community of practice. In fact, the economic valuation report that was prepared by Deloitte last year offered a preliminary estimate that the services our volunteers provide is worth nearly 1 million euros per year. We are so fortunate that longtime members of our community of practice are willing and able to share their knowledge, experience, and time with new members of the community, and we find that new members of the community follow suit and forward it on to the next group. We've had so much success in developing our volunteer network in the Asia region that for the last 3 years, the virtual Asia workshops have been taught by the Asia Regional Support Team and have been mentored largely by volunteers within the Asia region. And I've been thrilled to see how many people are here from TBIF in Taiwan. Two of them did a presentation in the last session. So many of them are here to support Spnhc and Tdwg, and so many of them are volunteer mentors in our network. GBIF's regional support teams are comprised of contractors who work part-time within the target regions to further assist the development of the community of practice, particularly in non-participant countries. These regional support teams complement the work done by the community volunteers and work in close collaboration with the nodes and elected regional representatives of the GBIF regions to strengthen the coordination of regional participation in GBIF. We have teams in Africa, Asia, Europe and Central Asia, and Latin America and the Caribbean. Our Asia regional support team, all 4 of them are here this week, and you can stop by and see them at our GBIF booth. The last area in our tools and approaches section is our training and learning resources. We want to ensure that there are opportunities for individuals to build those skills, which ultimately contribute to GBIF's work in the global community. In the previous version of the framework, we did not have a documented training strategy, but we do now. So in short, we strive to provide a blended learning environment that offers reusable and easily accessible opportunities for all levels in the framework. We strive to maintain a catalog of resources that further the goals to mobilize and use data, and we collaborate with a range of volunteers and experts in the development and delivery of materials. Our current course catalog includes these 4 courses, as well as a series of courses that are specific to nodes and those interested in becoming formal participants. The courses are maintained on GitHub, and we place continued emphasis on refining and updating materials as technology changes. And I'm rapidly running out of time. We also have digital documentation. It's intended to support our data publishing and use communities. The digital first documentation is on a range of topics relating to biodiversity informatics and open biodiversity data. We have 10 guides that have been developed by community experts since 2020, including Arthur Chapman, who worked on the Sensitive Species Guide and the Georeferencing Guide, among a couple others with georeferencing. Text and other content are maintained in GitHub repositories. Support for translations is integrated via Crowdin. In addition to our best practice guides, the GBIF Secretariat has recently launched our technical documents of GBIF tools and information. It is on a subsite of gbif.org, so techdocs.gbif.org. It includes the GBIF API, IPT, data processing and quality, using data, cloud services, system components, and more. And then the final element is the Data Use Club. We try to offer 2 seminars and 2 practical sessions each year, and we generally have between 50 and 80 participants at each offering. We also are partners in the Data Camp Donates program, and this year we have issued free licenses to 199 individuals, and the most frequently studied topics by these learners are R, Python, and SQL. So as I conclude this presentation and show you pictures of the people that make up our community of practice, I hope I have provided you with some examples that you may find adaptable to your own context. Participant nodes and other partners are encouraged to use this framework when designing their own capacity enhancement activities. And this framework is shared as a living digital document on GitHub, open for comment and suggestion, and reviews from the community at any time. And thank you for listening. And I didn't go quite over it yet. There are QR codes for the framework document itself and the repository on GitHub if you have interest in looking at it or forking the material there. So thank you. No time for questions, but we'll have discussion at the end. And next up is a video from Lindsay Walker. And she is not online, but we have Katie Pearson back here, also from Symbiota, to be able to answer any questions. Thank you.
Hi, everyone. My name is Lindsay Walker. Hi everyone, my name is Lindsay Walker, and today I'll be presenting strategies for capacity building in the SYM28 user community that I and my collaborators, Katie, Jen, and Samantha, have completed as part of our work with the SYM28 Support Hub. Here's a brief overview of what I'll be talking about today. I'll give an introduction about what SYM28 is, what is a SYM28 Support Hub, and then I'll dive into our capacity building strategies, and then we'll wrap up with some reflections on our work. If you're not familiar with SYM28 itself is open source software for creating themed data portals that help people share and actively manage biodiversity data. SYM28 was originally created as a biodiversity data aggregator, but over time it has become increasingly popular for use as a collections management system. Because it is open source software, anyone can go review and use the SYM28 code available in our GitHub repository. So these themed data portals often have user communities that coalesce around common goals. And typically these goals are related to the management, digitization, and data mobilization of natural history collections. These portals are usually taxonomically or geographically themed, although some are institutionally based and others are project-based. And if you're interested, the full directory of SYM28 portals is available on our website, linked at the bottom of this slide. In the early 2000s, SYM28 was originally just used by a handful of herbaria in Arizona, and there was no real interface or user interface of any kind. Fast forwarding 10 years into the future, SYM28 had a front-end user interface, and there was very high interest among the thematic collections networks that were funded by the US National Science Foundation to start using SYM28 portals as part of their digitization projects. By 2020, the SYM28 user community had grown exponentially. Then, in the last 25 years or so, SYM28 has been used to create over 60 data portals that we know of, and has been used by thousands of people in over 1,000 collections to mobilize 87 million specimen occurrences. And so all of this is to say that around 2020, it became readily apparent that there was a growing need for building capacity within the SYM28 user community. Due to the popularity of SYM28 amongst the thematic collections networks that I just mentioned, when Tdwg, the US National Resource for Digitization, applied for its third round of funding, the proposal included funds for a new domain of Tdwg called the SYM28 Support Hub. So the SYM28 Support Hub officially became the sixth domain of Tdwg in late 2021. As part of this funding, we are mandated to do some very specific things. The first thing is that we provide help desk support for SYM28 users. On average, since 2021, we have responded to 120 inquiries per month, or over, by the time you see this presentation, or well over 3,700 help desk tickets. We're also responsible for creating SYM28 user documentation, as well as a sustainable business plan, and then we're responsible for hosting webinars, trainings, and workshops to improve community capacity. If you write to our help desk or attend one of our training sessions, you'll be likely interacting with any number of the individuals on our team, though most commonly me, Katie, Jen, and Samantha. So what are some of the strategies we have taken for building up the technical capacity and social network of SYM28 users so that they can use SYM28 as a digitization tool as well as work together to this end? The first strategy that was established early after the Support Hub was formed was a monthly meeting that we refer to as the SYM28 Support Group. During these meetings, a demonstration-style training is presented on a new topic every month, followed by time for community discussion and questions to give our users space to interact with both us and each other. Each meeting is recorded and then distributed to the user community, and the recordings are posted on our website and our YouTube channel. The target audience for the SYM28 Support Group are typically portal managers, curators, and collection managers, but some content is presented that is relevant to data editors or public data users, which are typically researchers. This meeting typically takes place on the first Monday of every month and lasts about an hour. The demonstration usually takes up about half of the time because we try to provide ample time for discussion so that there's a little bit more of an organic feel to the meetings. We also use a bit of time at the beginning of each meeting to make announcements. For example, if other upcoming professional development opportunities or training events might be happening soon. The demonstrations themselves often feature user-suggested topics and focus on one tool or workflows that we're not presenting an overwhelming amount of information, and people are actually learning about the things that they want to know more about. For example, we posted demonstrations on how to do things like upload data to SYM28 portals and how to add images to occurrence records. Most recently, we held a SYM28 support group meeting on portal features affected by our latest major code release. To date, we've completed 30 SYM28 support group meetings that have been attended over 900 times, of which about a third of these participants are repeats, so they do tend to attend multiple meetings over time. The recordings are also quite popular, and we've had over 3,100 views on our YouTube channel for Symbiotic Support group recordings alone. Moving forward, we'll likely continue this format, and something we're thinking about doing more of is encouraging more community-led content to highlight the unique ways Symbiotic users have started using their data portals. Because all these meetings are documented, we've integrated them throughout our written documentation known as Symbiotic Docs as a complement to our YouTube channel. And so it's been really handy to be able to embed these videos straight into Symbiotic Docs to make both written and video formats as accessible as possible, all in one place. The second major community capacity building strategy we've established is our Portal Advancement Campaign series. These events aim to sustain and reinvigorate a given portal community's data management and sharing momentum. During these month-long events, we provide training on various topics. We assist with lingering data management tasks, and similar to the support group, we provide a forum for the community discussion for a specific subset of our user community. The portal advancement campaigns are generally, but not exclusively, for collections administrators. I say not exclusively because sometimes we'll cover topics where they might actually be more relevant to data editors or a public portal user. For example, the Lichen and Bryophyte portal communities jointly presented a special session on checklists, which are often used by researchers. And unlike the support group, the campaigns provide sustained, focused attention on one portal community for approximately 1 month. The format is that we generally host a series of 4 weekly virtual office hours with both synchronous and asynchronous opportunities for participation, and each meeting is recorded and shared with that specific portal community in a follow-up email. As part of each campaign, we facilitate community interaction so that everyone can meet their fellow portal community members, but also learn more about the Symbiotic Support Hub and the resources we've made available to them. We also like to spend time on training users how to take advantage of the data cleaning tools that are built into their portals, as well as provide support with data management tasks that can't be easily accomplished through the main user interface. For example, helping batch cleanup coordinates that simply ended up in the wrong hemisphere. Once data cleaning training is complete, we'll progress to data publishing. Because the Support Hub is an associate participant node of GBIF, we're also able to help with endorsement to get new data publishers online. So between that and teaching people how to use the built-in data publishing tools in their portals, the campaigns have been quite successful for getting more collections into larger data aggregators like the GBIF portal. Since we started this series, we've completed 13 campaigns for 15 Symbiotic portals. This includes hosting 58 office hours, with more to come. Many of our campaign participants attend multiple meetings and occasionally multiple campaigns. Unlike the support group, the recordings for these events are not publicly listed, but we do share them with the specific portal communities after each event. People do seem to go back and watch those meetings and reference them, and they're also very handy for sharing with new portal community members who join a portal after a campaign has been completed. So we have also organized in-person events to build community capacity. For example, earlier this year, we coordinated a data cleaning workshop with other collections management systems at IDWG's annual digital data meeting. And last year, we hosted a workshop centered around the digital extended specimen. The data cleaning workshop emphasized how to find and fix errors in your specimen data, whereas the extended specimen event was geared toward increasing community awareness and driving conversations. The data cleaning workshop was especially successful in building new community connections, as this workshop included 20 IDWG-sponsored data capacity fellows who received travel stipends to attend the workshop because they had a demonstrated need for this professional development activity, and they had never engaged in an in-person IDWG-sponsored event before. As a direct result of our extended specimen workshop, a novel workflow for linking specimen records and Symbiotic portals to genetic data in external resources was developed. And for the data cleaning workshop, a series of data quality toolkits were designed and made available for reference. Switching gears, our fourth major strategy for community capacity building within the Symbiotic community is to provide multilingual documentation and training opportunities for our users. This initiative is led by my co-author, Samantha Orellana, who is a PhD student at Arizona State University. Samantha's outreach targets collection staff and researchers who prefer Spanish language training. Similar to our other events, she's hosted virtual workshops focused on building capacity for collections digitization. She does make the recordings available, and she also engages with users through our help desk. At last count, Samantha has hosted at least 7 workshops and given 14 presentations in Spanish, an emphasis on outreach to Symbiotic users in Latin America. This strategy has allowed her to provide training to users from at least 17 different countries. Samantha reports at least 50 participants per workshop, or over 500 total, which demonstrates a strong interest in capacity building events from collections communities in Spanish-speaking countries. And I don't want to go into too much detail because Samantha will be presenting on some of this progress in a talk tomorrow in Symposium number 30. So please consider attending her talk, which will emphasize her work with the collections communities in Guatemala. To wrap up, we want to present a few reflections on our work so far. First, in our experience, it seems that offering both synchronous and asynchronous opportunities for engagement and training are very important. While many users are participating in our virtual events, many appear to be referencing our documentation, and this will only become more important as our user community expands globally. We feel documentation is critical to building capacity. We feel strongly that offering multiple communication channels for training are important, which is one reason why we don't just host meetings, but we also provide one-on-one training opportunities and support through our help desk. Importantly, providing these channels in multiple languages can significantly expand your reach as well. While we're able to offer virtual events most regularly, the in-person events have received very positive feedback, and they provide very focused sprints of engagement as well as additional networking opportunities to our community members. And then lastly, we found that building capacity within the Symbiota user community is as much a social endeavor as it is technical, which is why our events usually include unstructured time for more organic conversation. People don't just want to learn something new, but they appreciate it when that information is coming from a source they have a close rapport with. If you have any questions about what we've presented today, my fellow Symbiota Support Hub member, Katie Pearson, is also attending the conference, and feel free to chat with her or send us an email. I'm only here virtually, sorry, not in person, but you can contact me as well at help@symbiota.org. Thank you very much for your attention today.
I think we can take one question.
Katie, where are you? Hi, everybody. And so just very quickly, your perception, and Laura, this includes you too. I read something recently when we talk about community building, the difference between facilitating the community to be able to do something and rely on you as a help desk member. And it appeared that novices don't see themselves as necessarily part of that network. Like they come for help and they get it, but they don't necessarily see themselves as the one paying it forward. So it was interesting, Laura's comment, and then what we're all trying to do in different areas, some of us here, about this community building, is helping build a community where the people that come to the table recognize themselves as then paying it forward, the ability to become, whatever our tools or services that we're using, allow them to become sources of help as well, so that we're not like a single well point, right? So how do you think Symbiota is doing that in this case?
I think one strategy that we've taken is we try to be as helpful as possible, but at times we will say like, here is the documentation for that, and kind of like let them work through that. And that requires us to have very good documentation at the first part, but also to not just like, if someone is repeatedly coming to us for similar tasks, we're going to like encourage them to investigate doing that themselves. And we are there to help them along the way. But I think providing them with the tools to learn it themselves is one way that we try to do that.
I guess then to be clear is, how do they learn? How do we make sure we teach them? And Laura, you referred to this directly. How do we ensure that that person then also comes to see themselves as a resource for the next person who has a question? Because now they know the answer and have that skill you encourage them to pick up. So they don't always have to come to you or the same 2 or 5 people, right? The more people, the nodes in that network become access points for the knowledge.
Yeah, I don't have a particular strategy for that. I think that they gaining the skills to do so kind of empowers them to then pass that forward to the next people.
I have something I'd like to add, but I want to come back to it when we go to the discussion. Next up is Andrea Weeks.
I have to adjust it for Andrea height. All right. All right. I'm going to stick to my script so I don't go over. Okay, I want to thank the symposium organizers for giving me the opportunity to talk to you all today about this project. This is a collaborative project that I've been working on for the last year with a number of people. Just because I'm new to this crowd, I'll introduce myself. I'm Andrea Weeks. I am a plant systematist. I'm also a professor of botany at George Mason University, and I direct a mid-size herbarium at my university. Okay, so today I will describe the rationale for the CISRS initiative, its learning objectives, and the outcomes from the assessment of our 2023 workshops. Our take-home message is that we want to encourage other taxon-based professional organizations to adapt our curriculum for their needs because it is effective. And to that end, the QR code in our logo, which I have many stickers here that I will hand out to all of you, this will send you to a link of all of our project materials, and so you can fork it and play with it and make it your own. The CISRS initiative is a workshop and symposium series, and we designed it initially for plant systematists as well as herbarium collections professionals, and it has been funded by the U.S. National Science Foundation since 2023. Its mission is described by its acronym. It supports inclusive and sustainable research infrastructure for systematics. The workshops train people in biodiversity informatics tools that uncover people data in natural history specimen databases. This crowd doesn't need a primer on this, but in the context of a specimen label, people data refer to the people listed in the collected by field and the identified by field. We all know, particularly in this crowd, that that accurate people data in natural history collections databases can advance the history of science. It can absolutely improve systematics research, and especially our emphasis is enhancing the sustainability of the research enterprise of systematics by allowing collectors and identifiers to get credit for their intellectual work. So not just papers, but credit for the specimens and their identifications, so they can list that in their CV. Unique identifiers for people data, of course, they can be included in databases within Darwin Core. There are special fields for it, but the application of these unique identifiers requires community participation. And there is a very large need for awareness building across career levels. About the concepts and the tools and the practices that are required to leverage the benefits of these unique identifiers to realize all the benefits of using people data at scale. This is really the rationale for CISRS. Okay, so CISRS workshops last for 4 hours. Participants arrive having completed about an hour worth of homework. They watch a quick video, they skim a review paper, and they also create a login profile for Bionomia, which is the major tool that we learn, or the participants learn how to use. The first thing that participants do when they arrive is that they use Bionomia, they learn how to use Bionomia to attribute specimens to historical collectors. So these are deceased individuals, and the list that we have them work on is historical female botanists that were based in the United States, but you can choose your favorite group as you like. The next thing that participants do is they use Bionomia to locate their own specimens. And in doing so, they also are locating their annotations on the specimens. And in doing so, they build a profile page for themselves. This is just one example of a profile page. This page automatically maps the location of their collections, lists where specimens are deposited around the world, their specimens are deposited around the world, and reports the different scientific papers that have been published using their collections and their identifications. Then, using Zenodo, participants can also create a citation to their specimen-based work. However, as many of you are aware, Bionomia only sees what is in GBIF. And when participants realize this, it generates a lot of discussion, particularly for more established participants, those well into their career who have collected thousands and thousands of specimens, they realize that they can't find all of their specimens once they start looking for them because they haven't been digitized. And this leads to frustration, but then it quickly segues into really productive conversations about, well, maybe how can I help? Or what do you mean pushing data to GBIF? What does that mean? A lot of light bulbs get turned on. Okay, and lastly, participants watch a live demonstration of creating a new Wikidata entry for a historical botanist and linking this Q number into Bionomia so that you can start doing this history of science research about this individual. And this was an addition to our curriculum based on our first workshop, when from our feedback, we realized that a lot of our participants had their own people from their own collections that they knew a lot about but were hidden. They weren't out there in the Wikiverse. And so in each workshop, we work through this with a new hidden figure. In this case, this is just a snapshot of the story we tell about this particular individual, Lena Bondurant Henderson, who was a hidden figure in US botany in the early 20th century. But the storytelling is really engaging and people start sharing their own knowledge. Okay, we did assess the workshop and we used Qualtrics to do this. In the online application form, participants are informed about they are given informed consent about the ethical use of their personal data. And we do just collect very basic demographic data. The first assessment for the workshop is given directly after the workshop ends, before they leave. And then the second assessment survey is sent out 60 days later, just to assess just a little snapshot of the long-term impact. Okay, so in 2023, we delivered 3 workshops to 53 participants at 2 in-person conferences in the United States and 1 online Zoom webinar. Participants, their location or where they hailed from is located in this map across the United States, including Hawaii. And I will say we did have a number of international graduate students, but they were based at US institutions. We had 23 participants in the junior category. So these are what we call like early career individuals, like undergrads through postdocs, and then a senior cohort, including people with full-time jobs. These are either in the sort of the collections field or they were professors. From the first assessment, we learned that most participants reported very large learning gains in all of the informatic tools that they encountered, such as Bionomia. What is GBIF? What is Wikidata? What's an ORCID? What is Zenodo? And overall, junior and senior career cohorts had similar learning gains, except for 2 tools. The junior cohort had nearly twice learning gains for ORCID as compared to the senior cohort, and this is understandable. Most of the graduate students who took these workshops didn't have an ORCID, but we required them to get one in order to take the workshop. And then also the younger folk seemed to really understand what Zenodo was all about. And so they had significantly reported significantly understanding more about what Zenodo actually did at the end of the first workshop. The workshop did change behavior of the participants. After 2 months, when we polled them, it looked like, well, they reported that 73% of the participants reported using Bionomia after the workshop. And I think this is really striking because only 15% of our participants at the start of the workshop had any idea about what Bionomia was. They clearly found it a valuable tool and they kept working with it. Participants also reported having greatly increased their use of all the tools as compared to before the workshop. But I think the biggest surprise for me was the degree to which the workshop participants reported sharing what they learned with other people. So in the 45 respondents to the 60-day assessment that we sent out and then we got the report back, easily, like conservatively, they shared this knowledge with at least 100 other non-participants. Some people told 10 or more people, and they told their own botanical colleagues, they told non-botanical colleagues like entomologists and zoologists, and they also told their family and friends. So for instance, 93% of the workshop participants shared knowledge or skills with at least one botanical colleague. So you train one people and there is continuing dissemination for these tools. Junior and senior cohorts were largely similar in their behavior in terms of sharing what they learned with their friends and colleagues. But it looked like the senior career people shared more outside of their own taxon domain. And this makes sense. As you advance in your career, your professional network grows. So as a botanist, I know a lot of zoologists, but a graduate student might only know botanists. So our assessment results indicate that the format of this training is quite effective in meeting its objectives. Not surprisingly, and this is a lesson learned here, we found that we were able to offer participants stipends, and that is because we had an NSF grant. Like, we gave them money to come to this workshop. And when we polled them, we learned, interestingly, that 22% of the participants, it was definitive. The availability of money turned their head to sign up for this workshop. And they continued, they got a lot out of it, we know that, but it was just the money that led them to be interested in this workshop. And a very large proportion of people reported that this stipend was definitive in allowing them to attend an in-person conference. And I think this speaks to a large degree to the perceptual barriers that is out there in the community in terms of the importance of people data, but also the really big financial need that our community has to getting this type of training. So it's just something to think about in the future. And again, our take-home message is that we're really excited to share what we sort of have designed with other taxon groups. So the entomologists, the malacologists, the mycologists, you have your own separate communities, and there's every year a cohort of new scientists coming into the field who have no clue what GBIF is. And this idea of sharing knowledge through time, I think, is really important. We have published a workshop report in Research Ideas and Outcome. It is also linked to the QR code. And in my acknowledgements, I want to thank everyone for your attention. And also my co-PIs, Dr. Sean Krosnick and Dr. Twinell Majors at Tennessee Tech University, and a large cohort of senior personnel like Deb, David Shorthouse, as well as all of the workshop participants who provided really useful feedback, and the symposium attendees who also provided constructive feedback. So with that, I think I'm at time, and I'd be happy to take any questions. Thank you.
We will save questions for discussion time. And next up is Joanna.
Hi, so I'm Joanna, and I'm a Biodiversity Project Manager at the European Bioinformatics Institute, more precisely at the European Nucleotide Archive. And I'm here for something a little bit different from what's been discussed until now this afternoon in terms of scaling up data mobilization, because I'm going to present an ongoing project, which is called eDNA Aquaplan, one where an overview for eDNA sequencing, so environmental DNA sequencing of aquatic organisms and downstream ecosystem standardization is taking place. Here. So this project has been propelled by the need to integrate the eDNA derived data into biodiversity assessments and biodiversity monitoring for policy also. And we all know that eDNA data usage is scaling up, and to be able to unleash the power of biomonitoring and documenting observations in aquatic environments for example, the European Biodiversity Strategy and the Convention of Biologic Diversity, we need to integrate this data, and we need this data to be shared with rich metadata, properly documented, and in an interoperable way so that it can be highly reused. However, there's still some lack of standardization, and the eDNA application tends to be still a little bit limited by the lack of standardized formats and consolidated data repositories. And we believe by that this information exchange and collaborative efforts using eDNA-derived data will happen with the appropriate tools and necessary standards. So this project, which is a plan for an European eDNA digital ecosystem for next generation of aquatic biodiversity monitoring, has these three main goals, which is to identify related eDNA and DNA aquatic monitoring efforts and to document it so they'll get information on the standards being used, on the methods, on the workflows. And then finally assess the feasibility of building an aquatic eDNA ecosystem, so a digital ecosystem, and an integrated reference library to be able to foster its use. So eDNA Aqua Plan, as I said, is an European project. We have 18 partners from 11 countries. The project is a 3-year project and is now reaching the end of its first year. So it's in its beginning still. This is a general overview of the project. So there's a project coordination tasks, and then there are 2 main tasks that relate with auditing and assessing gap analysis in terms of standards and workflows and repositories for eDNA, and picking up that information towards documenting the standards and interoperability within that, and using use cases as of example data and workflows to inform on these standards and workflows and interoperability. And in the end, with the goal of building a blueprint and a roadmap for this digital ecosystem for eDNA, aquatic biomonitoring, and these integrated reference libraries. What I'm going to discuss today is mostly what has been done within the audit and gap analysis and how this can contribute to the standards and interoperability. These are just initial methods and initial results that we're having, so more information will come in the future. So for assessing the gap, for doing the gap analysis, the project has started by trying to mine eDNA repositories and inventories and communicating within the project partners and further on to try to assess the metadata and data standards being used and how this is reflected in the information that is being released in the public repositories. So there was questionnaires that were sent out to researchers, and there was a gathering of information within the public repositories. They identified first the different repositories that currently hold this type of information. And within the metadata that's being generally collected, they selected 40 attributes to analyze. So in the bottom, we have some initial results on the questionnaire survey that identify some of these metadata attributes and how they're connected to each other and how they are used in terms of quantities. And then on the left, we have an example of information being collected from some of the repositories being analyzed. And these include, for example, the 3 main databases within the INSTC, the International Nucleotide Sequences Database Collaboration, which include ENA, DDBJ, and NCBI. Also both OBIS and GBIF, large data aggregators, and other databases as, for example, EucBank, which is an European initiative for eukaryote information, in this case derived from eDNA, and MitoFish, among others. Part of this work has focused on getting data from the INSTC archives and looking into what's there. So one thing that was, this data was filtered based on sample metadata and for aquatic and environmental data. So we looked into sample and we looked into the packages and the checklists and how this is submitted within NCBI, DDBJ, and ENA, removing human information and looking mostly for data submitted within the MIXES, following the MIXES standards, which is these environmental packages and checklists, but also within generic checklists. Then this was filtered for aquatic and environment, and that's an example of different attributes that were taken in consideration for this filtering, and also according to certain library strategies more associated with eDNA. And this filtering being applied to samples associated with raw reads information, we got a total data set of 1.7 million aquatic environmental read records. One thing that we could take out of this exercise was that the querying of these databases was a bit complex. So to get this aquatic environmental data separate aside, we had to build a somewhat complex query to do that, which is something that I think we can improve. Looking into metadata in INSTC, we can also see that most, and this is listing, so different checklists available at ENA and NCBI, and the number of records associated with the checklists. And we can see in red that there are many records that are submitted into default checklists. And this is not so good because default checklists are minimum, so they require minimum mandatory information, and so we are probably missing a lot of relevant metadata in these records. There is a wide usage also of the MixS and other metagenome and environmental checklists in NCBI and Tdwg, but even so, there's a lot of information being submitted with minimum standards. And this is just another example of what's being done in terms of, for example, on the left, we see an example of metadata associated with experiments, so associated with the raw reads. And we see that most of the data comes from metagenomic library source, using amplicon methods, using Illumina platforms, and submitted within the last 5 years, which shows the increase of data being submitted through time, which is quite significant. And on the right, we see, for example, on the top graph, the distribution of these read data by country. And we see a good distribution, a lot of data coming from the US and from China, but also a big slice with still missing information. And we believe that this will reduce through time because INSTC made country and date information mandatory recently, so since last year. So we believe that more recent records will all contain, or at least most of them contain this country information. And in the bottom, there's also a distribution according to ocean, where we see a distribution that is agreeing with the above records on country. And also just some results, because I think I forgot to mention it, but there's also being used some large language models to text mine publications and to get information on metadata associated with this type of data. And this is just an example on freshwater types, where we see that most of the data is coming from lake and rivers, but also from other types of freshwater environments. So to summarize, we see, so the information that we are gathering in terms of this auditing and trying to identify gaps through these different methods and referring both to environmental DNA, but also to reference libraries and overall workflows will help us document the standards and current interoperability to try to improve them, both for these, so eDNA data repositories, genetic reference library, but also infrastructure, services, linking, and compatibility. And the project will use marine and freshwater use cases to inform on these standards and how this is working, with the main goal in the end to have a blueprint and a roadmap towards good digital ecosystem for eDNA and integrated reference libraries. So just like to thank all project participants, external advisors, my colleagues in MLEBI, particularly Peter that did all the graphics in the presentation, GBIF, and also the symposium organizers for presenting this work. Thank you.
We have time for questions.
Hey, my name is Rasa. already was here standing. And I spent many, many years working with DNA and especially eDNA. So I worked a lot with methods and workflows and implementation in the monitoring systems. But now I'm moving away from it, live. So my question is, so I'm still quite surprised that there is no standards. I mean, it's not a new method. It had been, I don't know, 10, 15 years ago, right? And we still lack of standards.
Well, there are standards, but they're not totally adjusted to what this method, so the type of analysis done. So there's still a few things lacking in terms of standardization. There's a lot of different ways of doing things.
Yes, exactly.
And that's not always documented and well documented in a way that can be interoperable. I think that's the bit that's missing. And then I think it's mostly the way that this data is made public, because you have, for example, INSTC for the sequence data. Not everybody submits to that. Yeah. And then you have, for example, GBIF for the occurrence data. Not all information goes there. A lot of information stands freely on other types of platform where the data is less interoperable. And I think that's where the direction that we're trying to take.
And I worked for that, mainly my projects were governmental. So all the data was kind of, in the report, somewhere end up in the drawer somewhere, right?
Yes.
And another question was, I was kind of sad, a little bit disappointed, as I'm from Sweden, right? Sweden is not a part of the aqua plan. Why?
I'm sorry, but I can't answer that.
And
so
also
I wasn't
that
a part of
Baltic
the
countries are also missing, quite a big part of the Baltic countries. Do you know why? Is it lack of sort of like spread of information? Because I know that people are working with it.
Yeah,
and it would be important to sort of include as many as possible. Do you have any ideas or like how to do it?
Honestly, I don't. I wasn't a part of the initial design of the project, so I'm not sure why not everyone or every country is included. I think, maybe.
You can.
Can contribute to that.
So I was involved in the previous project, Aqua Net. What was a cost action with only funding for networking and not for staff. And this Aqua Net plan is in the instrument of the EU that is called missions, where there is a much smaller budget than for other EU projects. So they had to make a selection on the partners. But it doesn't mean that others, for example, my institution, who was even work package leader in the cost action, is not in the Aqua plan because there was simply not enough money and it was not feasible. But it doesn't mean that we are not involved anymore. And we have worked a lot with Sweden in that project, so they can surely still be part, but not as a funded partner.
Yeah, that's what I'm going to say. I believe all project partners will appreciate contributions from other places.
A question.
Go if it's quick.
I will try. The short version of my question: are you developing a standard through ISO, or are you going to work through Tdwg? Is it going to be an open standard that everyone can use, or is it going to be a standard that people have to pay to use?
Definitely not paying. So I would say that everything that comes out of this project will be open. I think the first things that are being done are assessing what standards are being used and then make proposals. And there is collaboration with other projects to be in a collaborative process.
I asked because of an earlier experience with this program. I was understanding they wanted to go through ISO, and I had presented at a symposium they put together for DNA Aquanet, and I was like, make the standard open. So perhaps.
Okay, but I will pass on that information to the project partners, and we'll confirm if otherwise. Thank you.
Thank you. Okay, now we have our final talk with Miwa.
Hi everyone, my name is Miwa. Thank you for staying until the end of the day and then tuning into my talk. So my project that I've been working on the last 1 year and then for the next couple of years is to develop the best practice guidelines for formatting and publishing eDNA data according to FAIR principles. And then it's very related to what Johanna just presented. And she's actually one of the collaborators of my project. And we have a big group that I'm going to introduce later in the slides. So we have this FAIR eDNA working group. And I hope to answer some of the questions that was raised just now. So first of all, why do we need to make eDNA fair? And we all understand that there's a number of environmental stresses around the world, so we need a better level of biodiversity monitoring tools. And environmental DNA comes really useful for that. For some of you who might not be familiar with eDNA, so we collect environmental samples such as water, sediment, soil, and these days we can even do with air sample. We extract the DNA from those environmental samples, sequence them, and then we match that with the reference databases to understand what species, what these eDNA sequences are from what species. And in that way, we can understand what species were in that environment. And it's a really robust tool for environmental monitoring or biodiversity monitoring. And because of that, number of environmental studies are increasing very rapidly in the last 10 years. And this is a review paper we did on water samples on macroorganisms. So it's very specific. If we think about other type of samples like sediment, soil, air, on microorganism, number of publications per year could be well beyond 1,000. And each of these eDNA studies produce tremendous amount of DNA sequences, very valuable biodiversity data. And if we have all this eDNA data in a fair manner, what can we do with it? We can do all sorts of things, such as further studies at extended time and space. We can also increase the species identification accuracy by reanalyzing archived eDNA sequences with updated reference databases. And then we can enhance collaboration by reusing the data from different institutes, different countries and regions. And through these activities, we can generate knowledge at speed. And ultimately, we can support decision making. And at this moment, many of the eDNA data are unfortunately single use only. And one of the reasons is because it's sitting somewhere in this lost data map and it's not being shared. And some data are being shared, actually a lot of them. And there could be in GBIF, OBIS ALA, or NCBA, ENA, Tdwg, or sometimes in the dry adults or supplementary material of journals where there's open data repositories. Anyone can put anything in there with any format. And because of the format, unstandardized format, the data is shared but still not fair. They're still very difficult to collect them together and reuse them. And another reason why it's not fair is that data are archived, but then half empty, like what this title of the journal paper says. So people might share FASTQ files, the raw eDNA sequences, but without sample metadata. And those kind of things could be surprising for you guys here in the Tdwg conference, but it is very common for eDNA researchers or ecologists without any database background. To understand, okay, sample metadata is actually essential. We didn't know that, and we only submitted the sequence data. So in order to overcome these kind of challenges, we started this project to make eDNA fair. And this is the infographic of our project, starting from eDNA process, and then this data is at this moment, it's not standardized, and then those puzzle pieces cannot fit together. But we are working to change this data life cycle through these activities here. And then the outcome is that those pieces of puzzles can fit together, and it can also plug in with other type of biodiversity data, and then we can do all sort of cool things there. So I'm gonna walk you through this middle part here, what we've been doing for the last 1 year. So first of all, we established this FAIR eDNA working group. So collaboration is the key. It's a group of more than 20 researchers from multiple organizations, from multiple countries. We are a lot of eDNA researchers from multiple universities, and I'm one of them. My background is eDNA and marine biology. And, but we also have database managers from these institutions. So Joanna is one of them, and some of the collaborators are sitting in this room today. And we also have journal editors on board. So we started working with Wiley editors because they have environmental DNA journals, which is really relevant for this project. And the reason is because we want to develop these guidelines for eDNA data formatting, and we want to implement that into journal procedure so that this will be actually taken by eDNA researchers. And Wiley editors, they are really keen to work together with that towards it. So once we establish the guideline, we're going to try that out with the environmental DNA journals, and then we're going to spread to other journals within Wiley and hopefully across other publishers as well. And there's another collaboration opportunity that's really exciting. So our project is endorsed by United Nations Decade of Ocean Science. So through the program, there are multiple programs from around the world. So I have opportunity to connect with other researchers looking at a similar thing. So that's a really exciting opportunity for us. Now we're gonna get a little bit more into details, nitty-gritty of this standardization. So we're trying to standardize the data format and the vocabulary. And the main focus is so that the data is complete, not half empty. And then also they are machine readable, so we can tick the I and R. First thing we did is to define what types of data are important. And sorry, it might be really too small for you to read this, but we study metadata here and the sample metadata. They are essential. Then we divide this, the two major types of eDNA studies. One is the multi-taxa detection approach. So we basically sequence the data most of the time using metabarcoding approach. And then we get this species assignment at the end. But until we get to this point, there are multiple steps in the bioinformatics procedure. And it is very important for eDNA researchers, it's very important to understand what procedures were taken, because that could be very different depending on the study scope. And it's also different depending on the reuser's perspective as well, like what they need, what kind of quality control or assurance they need. So we want to make sure that we capture as much information as possible, and then we can filter data in and out based on the reuser's needs. And this is why we have these multiple layers, multiple intermediate files. We recommend people to format in this way and then submit. And then we have another type of approach, which is a target taxon approach. So this is just done by qPCR or dPCR. They don't sequence the data, but they can identify the presence, absence of a target species. And that's still very, very valuable biodiversity data. So we're also working really hard to capture this type of study. And so once this is formatted in this way, where to publish the data? So we're not really creating another database specifically for eDNA, because there are already so many out there. GBIF, OBIS, ENA, NCBI, they are working amazing, really hard, and doing amazing work to establish this platform. So what eDNA researchers can do is to submit this sample metadata and the raw sequences to NCBI, ENA, but then GBIF and OBIS, they're interested in an OTU table and this species assignment information. So those data, along with the metadata, of course, can be submitted to GBIF, OBIS, and then it can be publicly available and then fair in that way. Then what we did in the next step is to define, to develop an eDNA metadata checklist. So we use, this is a fraction of just a small part of the checklist table. Start from, there's different sections, start from a project information, it goes to sample collection, storage, preparation, all the way down to sequencing, bioinformatics, environmental variables as well. So now we have a big list, 275 fields. And when I showed this number, so many DNA researchers said, this is way too much, I'm not gonna do it, it's too much work. But I tried to convince them saying there's only 36 mandatory fields, so please do it. But then I got told just now that from other people that 36 is already too much as well. So we might have to review that and refine that and probably make highly recommended fields, mildly recommended fields, something like that. And but those mandatory fields are, for example, latitude, longitude, date, primers, project name. So it's very basic stuff that everyone has in their data set somewhere. So hope everyone can agree that this is important and not that hard to share. And of course, we went through Darwin Core checklist and then mixes as well, and then try to take as much existing terms as possible. And they have a very amazing comprehensive checklist. So that was really, really useful for us to start as a starting point. But we did identify there is some terms missing to capture unique characteristics of eDNA workflows and data types. So we did create some new terms, and that's how we ended up with 275. But those new terms, for example, so taxonomic identification is, I explained briefly earlier, but we match the eDNA sequences to the reference database sequences. And then the percentage match between those two are really important parameters for us to know how reliable is this taxonomic identification. So that kind of field was missing, so we included it. And then other thing is a contamination screening process. So contaminations can be quite problematic in eDNA workflow. So we have very robust procedures to monitor blank samples and then filter out some contaminations, non-target taxa, and so on. And those procedures were not captured by existing standards, so we created terms for that as well. Then we understand that data formatting can take time, and then time is the #1 limiting factor. So we are creating tools and scripts to help researchers to make the procedure quicker and more accurate as well. So for example, this one, eDNA FAIR metadata validator. So once people format the data according to our guideline, they can drop the data into here, submit, and then it will generate error and then warning messages. So those messages can be, for example, this is the mandatory field, but it's missing, or this is a fixed term, but then the entry is not from the fixed term options. So then the researchers can go back to their data, correct it, and then keep doing until there's no more error messages. So that's what we've been working on in the last 1 year. And what's next? So we want to publish these guidelines first as a preprint, and then so in that way we can spread the words, people can start using it, and we can receive feedback through those process. And we want to also integrate them into policy, as I mentioned about these Wiley journals. Then we want to establish an eDNA extension working with the mixes or GSC communities. Then we will keep developing formatting tools. So I show you one of the examples, but we have multiple ideas what kind of tools could be useful for data mobilization, data reformatting, and things like that. And we want to run workshops to eDNA labs and raise awareness about FAIR principles, FAIR values, because people, eDNA researchers, we don't really know what they are. We never heard of those terms, and we don't know why from the first place we should be doing that. So I want to change that kind of mindset through the workshops, and then also introduce details about these guidelines and help them to start working towards the FAIR practice. And we're going to continue working to revise and update the guidelines, because there's going to be some cases that maybe unique studies cannot fit into our format, and then we want to receive feedbacks like that. Then we're going to work together to revise it. We're going to implement some new technologies that we don't even know what's going to be out there in the next 5 years. We want to update that guidelines, implementing those things. We also want to actually reuse eDNA data for further studies and then do all sort of cool things. And finally, and very importantly, we want to work to harmonize FAIR and CARE so that we can make sure that FAIR data is reused in a respectful manner for indigenous people and local communities. And unfortunately, there was a CARE session at the same time as now, and then I missed it, but I would love to tune in later once it's available online. And also I want to connect with those people who've been working on the CARE principles during this conference. Thank you very much for listening to my talk, and then I have no idea how long I went for.
You're okay.
I hope I have some time to answer to your questions.
Is Peggy here?
Peggy's not here.
Okay, Peggy Newman would be a good person for you to talk to
She's
about the
actually
care. Oh, is she on your list?
here.
Yes, good.
Yeah, we've been talking about it a lot.
Okay, I think we can take one question and then we'll go to discussion. Dimitri, got your hand up.
Thank you. This is fantastic, and our discussion in the lunch and your talk combined made me realize one very important thing, and I just wanted to make it clear for the audience that working on publishing DNA-derived data through GBIF and OBIS, we started this 4 or 5 years ago, and there is a guide accepted by the OBIS and GBIF community. You can do it here and now, but that's publishing DNA-derived data beyond eDNA community to be picked up by people who don't work inside eDNA world or molecular biology. And what you're suggesting here is great, and the eDNA extension to mixes is very important. The deeper you index, this is a Tdwg trivia, the deeper you index, the more narrow is the scope of your data. The more shallow you index, the broader it flies. So I think our work, what we did for years now, and the work that you do in your project is very, very good match. So I think it's just important to understand the difference in kind of this potential reach of the data index deeply or shallowly. And as we are in the capacity session, it's very important to continue reaching out to actual audiences and address their needs. Writing the guide or writing recommendation, making a cookbook doesn't make people cooking. So you have to go there and actually do the training. It was fantastic to be in this session. The perfect closing. Thank you.
Thank you. Thank you for the great suggestions. And yeah, so we've been talking a lot about this project, and then we have Tobias and Thomas from GBIF as well, and they developed the DNA-derived data. Sorry, I didn't include that in my talk, but when I talk about the Darwin core and mixes, of course, I went through the DNA-derived extension, and then I took some of your new terms as well. And then, yeah, so whatever we create new terms, and yeah, we would like to talk about it, and then hopefully we can find a mutual way that we can work together. Thank you.
I would like all of our speakers to come up because we have 15 minutes still for discussion. Well, 12 minutes for discussion. So if our speakers would come to the front, and Katie, please come up too. And I do want to say that lost data map is my new favorite image. I love that. Yeah. Yes, come. I also, at the beginning of the session, I forgot to thank our sponsors, particularly our diamond sponsors. And I realized I also forgot to redo the instructions about the timer, but y'all figured it out, so good, thank you. And I really want to thank our technical support here today before everyone runs out of the room. So I want to have a bit of a discussion. I have some questions that I want to ask you guys, and then I have a couple questions for the audience, maybe. So which do you want me to start with? You guys or the audience? I'm not logged into the chat anymore. If you want to ask that, let's do that.
Yes, thank you very much for passing that on. So Esteban, it looks like you are still in the Zoom room, so if if you'd like, I could ask your question here now. Do you have a specific budget for the help desk capacity building strategy, or is a task done along with other Symbiota tasks?
Yeah, we haven't broken out our capacity building versus our other tasks budget-wise, but primarily the money that has come from iDig Bio is mostly community support and capacity. So you could probably say it's most of it.
Any other burning questions that you want to ask about the presentations before we? Behind you first, and then Deb.
So I was interested in the training for people-centered data, and one of the things that we have been trying to do is use identifiers more for people in the collections. We've heard an awesome presentation yesterday about Wikidata, there's ORCID ID, and there's Bionomia, and thinking about how all of these different parts fit together and instructing collections and users for that one field that they get recorded by ID or identified by ID, what should they put there, and kind of what is that pathway for how things fit together? Awesome if you have the answer, can highlight that.
So, yeah, thank you. So, as I understand it, it's Q number for deceased individuals and ORCID ID for living individuals. But Siobhan may have very strong opinions about this, so please chime in.
In the typical New Zealand way, Wikidata is the one identifier that rules them all, because the Wikidata item should actually have the QID, it's the QID, but if it's done properly, it should have an ORCID identifier for the living person already. Living people should, if they've got an ORCID, they should have a Wikidata item, particularly if they're collectors or identifiers. And if they're deceased, they're going to have a Wikidata item if you want to add them. So I would say it is the one identifier that rules them all. It is the ring, and it's the one you should use.
So I heard at one point that you're not supposed to make a Wikidata item for a living person. Is that false?
Oh, what a, no. I'm Wikifying as we go. This conference, I've done IBC, I'm making lots and lots of Wikidata items for living people. No, yeah. Yeah, even then, well, because it's conflict of interest, but even then, Wikidata, because it's a little more lax than Wikipedia, definitely don't do anything in Wikipedia about yourself, conflict of interest. Wikidata, so long as you've got a reference to support the factual statement you're making, you can actually, although there are people who argue against it, make a Wikidata item for yourself and add statements to it. Yeah. But yes, you can make Wikidata items for living people. You can add their ORCID identifier to that Wikidata item. It's the QID, it's the one thing that rules them all.
Okay, I'm going to suggest, Deb, you ask your question and we'll finish answering your question from earlier. And then just based on the time, the questions I have for the symposium discussion, I'm going to put them in Oxford chat and encourage y'all to answer those questions in the chat so we can keep the conversation going.
Mine's very quick. It's a general statement to think about for the last speaker. Excellent. I loved every part of it. What I want to suggest, the constant battle that you raised, that we have a long history of, people saying that's too many fields. We need them. If we're saying this is the kind of scientific questions we want to be able to answer, we need that data. So how can we make it better for the scientists to provide that data? The tools that you raised, how can we think more constructively about how to make that metadata collect much of itself? Just by the initial act of like creating a project. We're going to go here, we're going to do this. These are the people who are going to go. These are the techniques we're going to use. So you fill all that at the advance, and then when you get that sample and you pluck it out of the ground or take a picture or whatever, all that metadata comes for free, so to speak, right? So you're not filling out that metadata for each individual record. And that we are still needing to evolve a lot in that realm. Right? We still have people taking an image and then putting the metadata for each field for that image, rather than saying we're about to take 200,000 images and they're all the same from the same country, or they're all from the same collector, and adding all that metadata at one time to that giant pile. So you're building the tools. think hard about which of those fields could be applied in a broader sense.
Yeah, thank you. That's a great point. Yeah, so when I'm going to actively run a lot of workshop to EDNA labs to introduce about these guidelines, and then I've already started talking out in conferences and things like that. And what I always tell is when you start the project from the beginning of the project, use this format so that you don't need to reformat later when the study is about to publish and spend another day or two to do this, and people just get fed up with that. So yeah, so the advice is that, use this format from the beginning. And even if the data cannot be shared for any reason, it is a great advantage for example, just within the eDNA labs, if multiple students and then researchers use the same format, that's already a great advantage. And then they can use our guidelines for that. So yeah, hopefully people will start changing that behavior from the beginning of the project. Thank you.
I also really enjoyed your talk, and I was also overwhelmed by the 247. But I thought about what if you sort of gamified this and said, okay, gold, silver, and bronze level, and then everybody wants gold level. But so that the data, so in your lab you could be like, well, we have to collect a bronze level. Right? Or, or if you want to be a friend to your future self, you do gold standard. So 30 years later, you can go back and be like, what did I do? But, why bother? Why bother going to silver? Why bother going to gold? And that, I could wrap my head around that. Yeah. That's just a comment.
Yeah, that's a great idea. And it's like you're actually the second person who thought about it. And it was only yesterday, I was told by other eDNA researcher, why don't you have this kind of certification system so that labs are more aware about it and try to achieve the better standard? And it applies a lot, not just for the researchers, but sequencing companies. So they tend to hold on a lot of data unless the users pay more extra money to get, okay, I want the raw sequence data. The data should be there, but then the sequencing companies don't want to share unless people pay extra money. So we could use this kind of certification qualification to sequencing companies, saying okay, you provide all this data to the users, you can have a gold medal, and then something like that. So that in that way, it's for eDNA users, it's clear to find which sequencing companies to go with, because they just want to go with the good standard, good practice that they provide all the data sets. So yeah, that's something that came to my mind just yesterday, and yeah, it'll be really useful to work on.
I would like to challenge everybody in the room a little bit. Capacity is all good, and when you're in Tdwg, you think that everybody should be a Tdwgian, and I often agree with that, but maybe this time I'll disagree a little bit. And when capacity is mixed with recruitment, then you're assuming that everybody will be like you, waving arms and making new helpers. But capacity building, in my view, is a lossy process. You educate people, and your kind of default target that when people leave your capacity event, they should feel good and comfortable with what they are doing. They might not become helpers, and that's also right. So there are kind of capacity layers, and I think it's important that we accept these kind of differences at exit. And so I wouldn't expect full conversion, that everybody should be a helper eventually.
I don't disagree. I'm saying that there are strategies that help people see themselves as, and it's mostly when they're medium or expert level, then they begin to see themselves as ability to help others. Novices don't. They self-interpret that they don't have anything to add. We made a strong commitment at this workshop for CISRIS to explain to these people that now you understand bionomia and you understand something about the importance of Wikidata. You have the power to help your local community. You have. And that's something new for them. We gave them agency they didn't have before. That's what I'm saying. Yeah.
I would follow up that is exactly what we did in the bid program.
was give people that agency. We embedded the learning, the workshops in their program or their projects. It was a requirement that they send someone to the workshop. It was a requirement that that person earned the digital badge at either the basic or the advanced level. We made it a requirement that wasn't really a hard requirement, but that they needed to disseminate the information. They had to tell us how they were going to disseminate the learning when they got home. And we encouraged them to do it immediately after the workshop. So those people that did earn the advanced badge, so in the workshops, we also staffed them with mentors, and their mentors became their friends, and they're somebody to look up to in the community. The people that earned the advanced badge, we invited them to become mentors immediately. So even though they might be fresh and brand new, if they volunteered immediately for an event, we paired them with an experienced mentor so that they could learn. So we tried to build it into that. So with the BID project and the BIFA projects, it was easy because we could make those requirements. It's not as easy when it's not a requirement, but we try to do the same thing. The Asia Data workshops that they've done the last 3 years, they hold a contest for 7 to 10 people that are willing to publish a data set immediately following the workshop. And, they get some GBIF goodies. It's not a big prize. But people are excited to do that because to get their data up immediately. And there's a little bit of hand holding for the first publication, but once they've done it, then the next time they're on their own. They might ask for advice, but it's up to them to do it the next time. So we try to build it in that they do have the capacity to do these things. So we're at 5:30. I will add more discussion questions to the chat if anyone cares to continue conversations. Thank you so much to all the speakers. I really loved hearing all the presentations today. And I'd like to encourage everyone to go to the session 30, which is also on capacity. And I think there's one or two of them tomorrow. And it is on bridging communities and building capacity for digital collections mobilization. So a bit more focused on collections, but I will be at one of those sessions. So just to watch, but I encourage you all to go to that too. So thank you so much.